And so we're back, and we're going to enter this next section of the program in the subject matter, looking at autism spectrum disorder, some of the research in this field. Now, I'm not an expert in this field. I don't see patients that have autism. To me, it's a very ill-defined area of research. I think I've seen parents who've applied a lot of the things I'm going to talk about in a very aggressive way with their autistic children. And, the results really vary significantly to sometimes where there's no change, and sometimes there's a very small change. Sometimes the changes are more significant. It's very hard to predict. It's hard to know to what extent nutrition and lifestyle medicine interventions can really have an impact here. But I thought I would introduce you to the leading researchers in this field, the people that I have respect for, and show you some of the approaches that they use. And these are the more recognized practitioners in the world. So I'll show you the 10 novel approaches. If you're someone who's interested in this area, these would be the things I think to pursue in a more serious way. So to start with, food sensitivities. Many of the health practitioners doing work in this field who are legitimate, most of them agree that dairy and gluten-based products should be removed from the diet. This seems to be a consensus that most of the experts agree on. I don't know exactly why these foods are a huge irritant to brain function in these individuals, but there seems to be general agreement there. The second thing is that some of them also eliminate foods that contain salicylates. So, Dr. Feingold back a number of years ago identified that certain foods that naturally contain salicylates, and there are many of them, can actually create like brain fog and cause these problems that would aggravate learning disabilities and autism problems and so on. So I don't know enough about salicylates in food and the effect on the brain to be an expert, but some of these health practitioners who deal in this area of autism suggest that the autistic individual avoid foods that are high in salicylates. Then we come to dysbiosis, which is an interesting area. So this is about where you have a lot of unfriendly gut bacteria and candida, things that are growing in the large intestine that are crowding out the friendly bacteria. And these, the unfriendly gut bacteria and candida are secreting endotoxins that get into the bloodstream, and they cross the blood-brain barrier, and they disrupt neurotransmitter function in a significant way. Is one of the things that they do. And this can make autism much worse. So there's some validity here. Now, the reason why we know there's some validity to it is because there have been a couple of preliminary trials where they've identified that the individual has, say, a lot of Clostridia bacteria in the large intestine. And they've given the subject an antibiotic drug like vancomycin. And the vancomycin then kills the gut bacteria, or they found that there was a candida, so they gave them nystatin and killed off these pathogens. And you actually see significant improvement in concentration ability and normal functioning, sociability. You have the autism symptoms, are dramatically reversed. And then when the person goes off the antibiotic, the whole autism spectrum thing comes back to what it was before. So I think there's some validity here. So this is why I took this course by Great Plains Laboratory, Inc., because they have this test called the urinary organic acids test. And I think this is a weekend seminar worth taking if you want to do some continuing education. Because with this assessment called the OAT, the organic acids test, it looks at end products of metabolism found in the urine. It's the first urine sample of the day. And so they can find these sort of toxins that are released by these different bacteria and fungal species. And those endotoxins get into the bloodstream, cross the blood-brain barrier, and can disrupt neurotransmitter function. So an example would be certain types of Clostridia bacteria that are not causing a serious infection where you'd be sick and throwing up and fever, just low grade. They secrete this HPA compound, an acid, which is found in the urine. And it's found in high concentrations in the urine of individuals with autism spectrum disorder and schizophrenia. So it's sort of a biomarker, if you will, that there's some bacterial infection there on a low grade. And if you could use probiotics or prebiotics and other strategies to change the microflora, that you might see a decrease in the presence of HPA and an improvement in symptoms. And that has been shown at times. And Clostridia toxins also create endotoxins that increase dopamine, and if there's too much dopamine, it creates an imbalance with neurotransmitters and can actually disrupt behavior. And then when the Candida toxin can inhibit, look at this, the toxin from Candida can inhibit the 5-methylene tetrahydrofolate reductase enzyme. So the same way that you can be born with a genetic defect where you can't make the 5-methylene tetrahydrofolate reductase enzyme, and so then you can't make 5-tetrahydrofolate, so then you can't make S-adenosylmethionine very well. But the same thing can happen just with certain toxins that inhibit the enzyme. So without enough S-adenosylmethionine, because you're not converting homocysteine now to methionine, then you can't make neurotransmitters effectively. And detoxification becomes sluggish, and you start to even have defects in glutathione synthesis. So all of this leads to a way to identify these biomarkers, and some of these biomarkers also identify, end products in the urine that would suggest mitochondrial dysfunction. So this could be important for someone with Parkinson's disease as well. But in autism, mitochondrial dysfunction is one of the things they suggest is part of this, as is glutathione deficiency and a lot of free radical stress going on in the brain, and imbalances of neurotransmitters. And the person has chemical toxicities that need to be chelated or taken out of their system. So the claim is that the urinary organic acids test is a more reliable way to look at stool cultures, or to look at for pathogens in the large intestine. That would be a superior way than looking at just stool cultures if you're looking for low-grade pathogens in the gut microflora. But the traditional way of looking for different types of bacteria and fungi in the gut would be to look at stool cultures and see what's there. But the proponents of the urinary organic acids test say that this is a more sensitive test, and it actually looks at physiological function as well. It's not just looking at the bacteria only. Anyway, it's a fascinating seminar. I would suggest you look at it, because it has application for other health conditions: Alzheimer's, Parkinson's, any type of oxidative stress-based disease. I think it's a useful indicator so you can monitor the person's improvement. And then the fourth hypothesis is about leaky gut and the serum zonulin test. So the thinking is that autistic subjects have increased gut permeability. It allows certain peptides to cross from the gut into the bloodstream, and then antibodies interact with it, producing these immune inflammatory responses. Some of these occur in the brain, so you get neuroinflammation. And neuroinflammation has been shown to be a factor, actually, in autism spectrum disorder. So this was all sort of theoretically based, and then someone came along and created the serum zonulin test, which actually looks at, does the person actually have leaky gut? And if they do, often it's due to inflammation from the intake of gluten foods, as you're going to see here. So in recent years, scientists have identified zonulin as a key biomarker for intestinal permeability, which has been associated with celiac disease and non-celiac gluten sensitivity and other GI and systemic conditions. So when we talk about gastrointestinal health, and one of the subjects here on the platform, we'll spend a lot of time with celiacs and non-celiac gluten sensitivity. But the human zonulin is a protein that increases intestinal permeability in the small intestine, and it participates in intestinal innate immunity as well. So circulating zonulin in the serum is considered a very useful marker of intestinal permeability. And that's because in the healthy GI tract, the tight junctions between the cells prevent the unregulated influx of luminal contents. However, certain situations, such as the presence of gluten for people with celiac disease or non-celiac gluten sensitivity, can lead to high levels of zonulin in the GI tract. And this can induce the breakdown of these tight junctions, leading to intestinal permeability, allowing zonulin to go from the gut into the bloodstream where it can be measured. As a result, circulating zonulin is a clinically useful marker of intestinal permeability. And the other thing is that because zonulin is the only regulator of intestinal permeability known to be reversible, it makes it a valuable monitoring therapeutic intervention as well. So if you start to heal the gut, then when you retest the serum zonulin, the levels should be lower, so that as the gut starts to get sealed off and you heal it, zonulin doesn't leak into the bloodstream, so you know that you're getting improvement because the zonulin level in the serum is coming down. But several autoimmune and inflammatory and neoplastic diseases have been associated with elevated levels of zonulin as well. And there's evidence that increased intestinal permeability occurs in those conditions, which can be identified with a serum zonulin test. So leaky gut is also one of those things that gets kicked around in the autism discussion. So doing the serum zonulin test, taking the person off of gluten and then seeing if the zonulin comes down and you're improving the sort of gut function, it's sort of a marker to look at that. And there might be other foods as well that are damaging the gut, and we'll look at that when we talk about food sensitivity testing. But the next thing that comes up a lot in these autism discussions around with these researchers is methylation reactions, and they suggest that people with autism may be more inclined to have this methylene tetrahydrofolate reductase enzyme deficiency and methionine defects, where they can't make methionine that well, so they don't make S-adenosylmethionine optimally. And so with that, you're going to get a rise in homocysteine. So you should look at blood homocysteine in these individuals, as well as the red blood cell folate, the serum vitamin B12, maybe methylmalonic acid to look at how vitamin B12 enzymes are working, or to do the nutrigenomic testing to see if they actually have a 5-methylene tetrahydrofolate reductase enzyme genetic defect, which would really show up on nutrigenomic testing. But remember that these methylation reactions from SAMe are required to make neurotransmitters, to make phospholipids on the nerve cell membrane, for detoxification, and for sulfur-dependent detoxification and glutathione status. It's all tied in together to these methylation pathways. So, and the suggestion is that people with autism have faulty pathways in this area. And so they're not making neurotransmitters effectively. The cell membrane is defective. Their glutathione status is down. They don't detoxify as well. They have more oxidative stress. And if they have more oxidative stress, one of the things you can that can identify that is to look at the urine and blood levels of methyl diamelinaldehyde, which is an indirect marker of free radical damage. We spoke about that in the previous course. So the urinary organic acid also helps to, that testing also helps to identify glutathione deficiency or defective glutathione function. So that's the other thing about the oat that I think makes it valuable for these patients. So just to revisit glutathione synthesis again, in our homocysteine methionine cycle, of course, folic acid and B12 are required to convert homocysteine to methionine in these methylation reactions. But once you have homocysteine, remember, with vitamin B6 becomes cystathionine, and then with vitamin B6 again, these are the transsulfuration pathways now. You form cysteine. And then with cysteine, with the help of GCL, this enzyme, it can add glutamate, right, to form glutamate with cysteine. And then with the help of glutathione synthase, you add the glycine and you form glutathione. So you remember that glutathione is cysteine and glutamic acid and glycine. It's a tripeptide. But you need these enzymes working properly to make it, and you need the vitamin B6 dependent enzymes here working properly in the transsulfuration reactions to make enough cysteine in the first place. So what we see in autism, this is what they suggest, is that there's a glutathione deficiency. These pathways are faulty in some way, as well as the methylation reactions going on to make S-adenosylmethionine. So just to look at that again, here's homocysteine again becoming methionine with the help of folic acid and vitamin B12, and then you have all these methylation reactions occurring to make neurotransmitters and DNA methylation and for detoxification, for creatine, all the things we've spoken about. And then homocysteine with the help of B6 becomes cysteine, and then cysteine picking up glutamic acid and glycine becomes glutathione. Remember that cysteine also can give rise to the amino acid taurine, a very important amino acid that we're going to see in the heart muscle, and may play a role in detoxification as well. So that's how you're making glutathione. And then glutathione is a fantastic antioxidant, so it can donate hydrogen electrons to quench free radicals, and in doing so, two glutathione molecules that have lost these hydrogen electrons then with a disulfide link sort of join together to form this. This is the oxidized form of glutathione, and it's represented as the GSSG. When you see it written that way, you know that's oxidized glutathione. Now you remember that alpha-lipoic acid can regenerate glutathione back to its antioxidant state, but in autism, what you have is a buildup of the oxidized form of glutathione, which can no longer act as an antioxidant, and you have a decreased amount of the antioxidant form of glutathione, which is the reduced glutathione. So once again, we see the formation of glutathione here, and again, it gets oxidized when it's quenching free radicals. The thing is, you have to be able to recycle it back to its antioxidant state. And in autism, there seems to be, for some reason, a defect in that process. So this study in 2009 shed some light on that. They looked at plasma biomarkers of oxidative stress have been reported in autistic children. And so they took these lymphoblastoid cells derived from autistic children, and they took some from unaffected individuals as well who were normal, and they were used to assess the relative concentrations of reduced glutathione and oxidized disulfide glutathione in cell extracts and isolated mitochondria to measure sort of the antioxidant capability. The results indicated that the reduced to the oxidized ratio was decreased, and the percentage of oxidized glutathione increased in both the cytosol and the mitochondria in the autism lymphoblastoid cells. So the autistic children showed evidence that they had more of the oxidized form of the glutathione and much less of the antioxidant reduced form of glutathione, which had been suggested over the years, but studies are now showing this in actual cells that are derived from these individuals. So then they challenged it in different ways, but the results suggest that autism lymphoblastoid cells exhibit a reduced glutathione reserve capacity in both the cytosol and the mitochondria that may compromise antioxidant defense and detoxification capacity under pro-oxidant conditions. So this is where the research is. I don't know how meaningful that is. I think we're in the early stages of understanding all of that. The sixth hypothesis is that you actually do take stool samples from the autistic individual, and you look for parasites, and you look for the types of pathogenic bacteria and yeast that might be there, and decide if you want to give them an antibiotic or probiotics, prebiotics, some combination to try to improve the gut microflora. So it's similar to the approach of the OAT, except you're using actual stool culture assessment. Then we have this new research around the endocannabinoid system. So these cannabinoid compounds that come from marijuana or cannabis can interact in the central nervous system. There are many endocannabinoid receptors, as you probably know. There's different types of them. And the different cannabinoid compounds can actually modulate through these receptors neuroinflammation. And we know that neuroinflammation is a key aspect in the autism brain. So the original research, or at least what's experimental at this point, is that the CBD component of cannabis, the cannabidiol, seems to have the greatest impact on modulating neuroinflammation. And the good news is that the CBD component of cannabis is the part that doesn't cause any psychoactive effects. The person's not getting stoned or not euphoric. There's no change in sensation. That the THC is what causes those psychoactive effects, but it's the CBD component of cannabis that actually has this effect on the receptors that modulate neuroinflammation. So it seems like a pretty safe intervention to try. So there's this really good 2017 review paper on this subject, the endocannabinoids in autism spectrum disorder, that you might want to read. The link is right there, and it just goes through the whole etiology of autism spectrum disorder that's not fully understood. There's involvement of genetics and environment and autoimmune factors, that there's an interplay of those things, but neuroinflammation has attracted a lot of attention because it seems to be a significant aspect of autism. And there's a growing number of medicinal benefits that we've been looking at when you modulate the endocannabinoid system, such as their ability to regulate neuroinflammation, neurogenesis, and memory. It raises the question as to whether or not there's some potential role here for certain elements of cannabis to modulate those receptors as part of the treatment for autism spectrum disorder. It's in the early stages. I would say keep your eyes open for the research on this as it unfolds. And then, of course, there's evidence that there's neurotransmitter imbalance in autism. And again, the urinary organic acids test by Great Plains Laboratory can actually help identify where those neurotransmitter imbalances are and possibly what's causing them. That's the thing that I like about that test. It gives you strategies that can help you then put in place dietary and supplement interventions, and sometimes the requirement for prescription antimicrobial drug therapy to actually improve the gut microflora. And then as you follow the person forward, you see a change in the end products of metabolism in the urine, and you see neurotransmitter balancing out in a way that seems to be beneficial for the patient. And then we have the whole area of food allergy testing. So, you have an allergist who does standard food allergy testing, and they say, well, I don't see any food allergies. But then you have sort of in the holistic world of nutrition, you have these people that do food sensitivity testing. And there's two main ones. There's the LCAT test, which is the most famous one. You can read about it here at this particular link. And the more recent one is known as the MRT, which is the mediator release test. And both of them are really looking at volume changes of certain immune cells when the person is challenged with certain types of agents. You look to see how the immune cells are responding in terms of their volume and shape and cytokines and things that they're releasing. And it tells you if the person has a sensitivity to a certain food. And some of these foods may actually cause damage to the gut lining and lead to leaky gut syndrome, and then you start seeing a rise in serum zonulin. You see how these things all dovetail together. But there's sort of these more sophisticated types of food sensitivity testing that some of these practitioners use and feel that this is the right way to approach what foods the individual should be sensitive to and which ones they're not sensitive to. And you have some type of objective measurement here using both the LCAT test and the MRT to help determine which foods are the ones you're sensitive to. So in someone that has these ill-defined things like chronic fatigue and lingering fibromyalgia type symptoms, and they seem to be doing everything right, and people with autism, maybe learning disorders, it might be worth going down the road of looking at these sort of food sensitivity tests using these sort of non-traditional ways outside of standard food allergy testing by an allergist. These are sort of in the world of more holistic medicine approaches. And they might be a worthwhile thing for certain types of patients. I wouldn't disagree with that. And then the 10th thing is this whole amino acid profile, where there are different practitioners who access different labs, and the labs look at your blood and they look at the amino acid, the free amino acid pool in your blood, and they look for imbalances. Then they give you a cocktail of amino acids to take orally that helps to balance out your amino acid profile. And some of the patients report improvement, with cancer and all kinds of conditions. It's unbelievable when you read some of the testimonials. I don't know what to make of it. I will tell you that I've had a couple of patients who had kids whose their children were sort of on the autism spectrum disorder. They weren't severe autistically challenged individuals, but they were at least moderate. And they take them from one doctor to the next to see if somebody has an answer. And someone who I know quite well, who had been a patient of mine, took his son to one of these natural practitioners who did the amino acid profile and created this amino acid cocktail for the kids to take. And the parent, who I know, was extremely skeptical, was expecting nothing to happen. So it wasn't like they were looking for a solution that was about to happen. They weren't anticipating anything good. But he said to me, it's been remarkable, the change in his behavior pattern. So, I then contacted one of these amino acid profile labs to try to understand what the science is behind this. And I will tell you that they gave me nothing that sounded scientific. I can't even recall what they said. It was just such mumbo jumbo. It just made no sense whatsoever to me. So I don't know what to make of it. All I can tell you is that some not just natural, some medical doctors who are involved in autism work like to have this amino acid profile done and get a specific amino acid cocktail and make it part of the nutritional protocol, and they claim that in some cases it's very useful. All I'm trying to do here is to bring these things to your attention. I'm not trying to endorse any particular method. I just want you to see these are 10 of the more novel strategies that seem to have some enough support behind them by the practitioners who are doing most of the legitimate work in the area of autism, that if you wanted to then pursue some of these intervention tools and assessments, I think these are the ones worthwhile looking into. And there's this fabulous little section in Dr. Jeffrey Bland's book called the book's called The Disease Delusion. And in the hardcover version, from page 57 to 63, he has a whole section here on why autism seems to be increasing across the population, largely due, it looks like there's been such a change to the environment in the last 50 to 70 years with the foods that we eat, the environmental exposures that we have, the amount of stress and sleep behaviors, that it hasn't affected our genes per se, but the epigenetic layer above the genes responds to these things, and it then directs the genes to do different things. So the same way that software tells the hardware what to do in a computer, the epigenome layer is instructing our genes to do different things than they did before. And we think this is one of the main reasons why autism has increased so much in recent years. So on this last slide in this section, I wanted to introduce you to 4 important leaders in the medical world of integrative autism treatment methods. These are the 4 people who I think, if you're going to follow the work in autism, I think these are the individuals who have really committed their lives to it, who seem to have the most legitimate information to share. This is who they are, where you can find them, and what they've done up till now, some of the things they've published. This was the fellow who I saw from Great Plains Lab who taught the course on the urinary organic acids test, Dr. Kurt Wohler. He was extremely knowledgeable. He's a very, very experienced practitioner in this area. And then you have R.H. Sandler, who did the original work on vancomycin and autism. You'll want to see some of the research that was published there. And then you have this individual who's the director of True Health Medical Center in Naperville, Dr. Andrew Usman, MD. You can see an interview with them here, but there's this fabulous video that they produced here that really, where they sit down and try to explain all of this stuff around methylation and glutathione and what they do with these patients. If autism is an area of interest for you, I think these are the individuals that you might want to look up and follow. So we'll leave it right there, and I'll come back and we will see you soon. Thank you.